It is now my proud privilege to introduce our next eminent speaker of the day, Dr. Shariq Naeem sir, a renowned clinical researcher with a passion for teaching and genomic-based research. Dr. Naeem holds several degrees in pharmacology and clinical research, including MBBS and MD from JNMCH AMU Aligarh, DNB, DM from AIMS Delhi, Diploma in Clinical Research and Pharmacovigilance, and PG Diploma in Hospital Management. Throughout his career, he has published numerous articles in national and international journals and has presented his work in various conferences worldwide. He has also been invited to deliver guest lectures at several prestigious institutions, including the Apollo Health Research Foundation. Dr. Naeem's expertise in pharmacology and clinical research is evident through his innovative work in designing a mannequin for teaching the roots of drug administration and developing 2 Android-based mobile apps for the assessment and reporting of adverse drug reaction in patients. His keen interest in personalized medicine, drug discovery, and preclinical research has led him to be a valuable member of various institutional committees. As an assistant professor at the Department of Pharmacology at JNMCH AMU Aligarh, Dr. Shariq has been instrumental in conducting various conferences and workshops, and his dedication to improving medical education is truly inspiring. Today, we have the privilege of listening to Dr. Naeem's insights on oncology clinical trials. Please give a warm welcome to Dr. Syed Naeem, sir. Over to you, sir.
Okay, thank you for that intro. So the first point that comes to mind is why oncology clinical trial are important in a workshop of methodology. So there are two major things why this is important. The first one is that the oncology clinical trials, most of the clinical trials which are happening around the world are of oncology origin. And secondly, most of the newer methodology which is coming up in clinical trial is basically of oncology origin. Therefore, if you want to study about the clinical trials, the methodology of the clinical trial, then it has to be oncology clinical trial that you have to study. So in this presentation, we're going to talk about 3 very important phases. First is the phase 0, then phase 1 and dose escalation studies in cancer patients, then cohort expansion, and phase very important, very unique phase 2 and phase 3 designs of oncology clinical trial, which is basket and umbrella trials. Then we'll have a brief look on what is N of 1 studies, and finally we will have a brief go on regulatory issues and approval guidelines. So before we go and study about the different methodologies of oncology clinical trial, we should understand about the different endpoints, because we are going to study these endpoints. We'll be discussing these endpoints, and we should have a knowledge about these endpoints. So the first one is the overall survival. This is the gold standard of all the endpoints. Basically, it means that when the patient is randomized, then till the death of the patient, this is the overall survival. This is the total survival time of the patient, and this is called as the overall survival. This is the gold standard. But the problem with this is the death of the patient. It takes a long time. You can have a 5-year survival rate. You can have 10-year survival. So it will take a very long time, and it is not feasible for the clinical trials to go on for that long period, but it always remains the gold standard. So what can we do? We can have some surrogate markers. So what is a surrogate marker? If we surrogate marker is basically a marker which is a proxy to a gold standard marker. Or basically, we want to decrease the time of our study with the help of some proxy marker. For example, in this case, the death is a proxy marker, and we are talking about the disease progression as a proxy marker. That is the gold standard. So, if we take another example, myocardial infarction is the gold standard or the clinical marker. and the calcium scores or the level of cholesterol can be your proxy marker, which is called as the surrogate marker. So now, if we take the disease progression, this marker, this endpoint will be called as progression-free survival. So the patient was randomized, and the time at which the disease was progressed or there was a death, this is called as the progression-free survival. If there is only the disease progression was the endpoint, then it will be called as the time to progression. So these two markers, the progression-free survival and the time to progression, they are the one which are very commonly used in the clinical trial, in the cancer chemotherapy clinical trials. These are the very commonly used markers. Now, this is the disease progression. What about this is the complete response. What about if the patient does not have a complete response? So the patient has partial response. What do you mean by partial response in a cancer patient? That it's the cancer shrinks in size. So it is not completely gone, but it shrinks in size, and we can have that partial response, the percentage of the size of the cancer that is shrink after a particular amount of time. So this is called as objective response. There's one more thing. What if the drug cannot decrease the size of the tumor, but it can inhibit the further increase of the tumor? So if the further increase of the tumor is decreased, then it is called as the clinical benefit rate. It's called as the clinical benefit rate. Now, the last endpoint is basically if the patient has a complete response, like in these cases, the patient has complete response. So in this case, the patient has a complete response. So how does we go about it? So what the patient can have recurrence. So we have to find out the endpoint for the recurrence. So in this case, the patient is randomized at the time the patient has complete response, and the time of recurrence is taken as the endpoint. This is called as the disease-free survival. So if we can understand these six endpoints, we will be able to understand the oncology clinical trial, the endpoints in the oncology clinical trials. Okay. So let's start with the phase one. Phase one trial, basically in all the clinical trials, the basic requirement or the outcome of the phase one trial is basically the DLT. What is DLT? It's the disease limited toxicity. So we want to know what is the disease limiting toxicity in a patient. And in the patients, for example, in the phase one, we are taking cancer patients, or in normal clinical trials, we are usually using the healthy volunteers. So for phase one trial, extensive preclinical data is required. We need to know what is the side effects of the drug, what are the efficacy endpoints of the drug, what is at what dose the disease has controlled in the rats or in animal population. And we can also measure any other in vitro tests for teratogenicity or carcinogenicity. We can do all these tests, and this extensive data is required. The FDA actually requires very extensive data, and therefore preclinical trial, they have to be very, very thorough. And after that, for the design of phase one trial, in cancer patients, we can do also pharmacodynamic studies. In normal phase one trial, we usually do pharmacokinetic studies because we are not having patients. But in phase one trial in cancer patient, because the drug is very toxic, we have to give it in the patients only. So in those patients, we can also do pharmacodynamic studies. Usually 20 to 30 patients in phase one trial, maybe more, but usually 20 to 30 patients are used in the phase one trial. The duration of this, the phase 1 trial is basically we want to give multiple dosings in each cycle, and the disease progression and or unacceptable toxicity is recorded. Then finally, the endpoint is batch suited drug, and the time for completion, which is normally 9 to 18 months, is also recorded. So please remember this infographics, because we are going to compare this infographics with the phase 0 trial as well. So how do we go about these phase 1 trial? So we do dose escalation studies. So what does a dose escalation study mean? It means that we start with the lower dose and slowly we increase the dose as and when we have experience at that particular dose level. So we have two kinds of dose escalation models. One is the rule-based model, and the second one is the model-based models designs. So in the first one, the rule-based model is basically a traditional 3+3 design. What it means is that we'll start with the lowest dose. This dose is basically HED. What is HED? Is the human equivalent dose. So we have find out from the extensive preclinical studies, there is some dose that we find out that it will not produce any adverse drug reaction, but it will produce very good clinical effects. So based on that, we will define the HED, human equivalent dose. After the HED has been defined, we'll start that with the HED or lower dose of the HED. And this is the starting dose. We'll start with giving this dose to 3 healthy volunteers, or in case of cancer trials, we'll give it to 3 cancer patients. And slowly we will titrate it upwards. We'll give this to different cohorts. And at the time when we get a dose-limiting toxicity, for example, out of the cohort of 3 individuals, we get one participant having this dose-limiting toxicity. So at this point of time, we want to increase our knowledge of this level, this drug level. So we'll again give this dose level to 3 more patients to see whether this dose is okay or not. This expansion of the patient, or this increase in the number of this patient at the same dose level, to know what is the toxicity level of this drug at this level. This is called as the expansion cohort. So this is called as the expansion cohort. We are expanding our knowledge. We are expanding the number of the participant at this dose level, and this is called as the expansion cohort. So once we find out that out of 6 individuals, 6 participants, we have toxicity for 2. So therefore, we will go further down, and this will be the recommended dose for phase 2. This is called as the traditional 3+3 design. This is basically a design of for rule-based design. Now we'll take the example of pembrolizumab. This is a wonderful drug because this drug has all the ingredients, all the examples. It can fit in all the examples of cancer chemotherapy. It's an orphan drug for advanced melanoma patients. So it was approved as an orphan drug. Now, in case of phase 1 studies, we can see here that we are using the incremental doses with the help of Fibonacci series. So what is the Fibonacci series? Is basically we have to add the two numbers before, and it will give the answer to the third number afterwards. So that if we see with an example, we have first number 0, the second number is 1. So the third number will be 1 plus 0 is 1. Then 1 plus 1, 2. Then 2 plus 1, 3. Then 3 plus 2, 5. Then 5 plus 3, 8. This is the incremental ratio. So initially the incremental ratio is high, and you can see here that the incremental ratio goes down. This is the modified series, and this is again different series which are available. So the incremental ratio is high at the start, and it will go down as we increase the dose. So this is this is better because we want to test the higher doses in a very small increments, so that there is no overdose toxicity for the study participants. Next is this is rule-based models. So we have talked about the 3+3 design. Then the second model is the up and down model, in which one patient is given the drug in each cohort, and we will go up until we have a dose-limiting toxicity. Then we go down until we have no dose-limiting toxicity, and then we again go up and down until we reach a dose where the dose-limiting toxicity is the least. That is the recommended dose. In accelerated titration, this is done in the one patient only, the single patient, and will give incremental doses until we reach a DLT. Once we have reached a DLT, we'll try to increase the number of the patients here. We try to expand the cohort, giving it to 2 and 3 patient cohorts. And once when the DLT is achieved, then we'll say that this has the recommended dose. In pharmacology, what we are doing is we are using AOC from the preclinical studies. Remember, we said that we need the extensive data. So this is the extensive data. We are using AOC so that the pre-specified threshold is measured. And once we'll start with the one patient, and once that is reached, the pre-specific value is reached, then we'll again go for the three participants. And again, then it will be same as the up and down or the standard 3+3 model. Now, the other models are the continual assessment method and escalation with dose control overdose. So the major drawback of the rule-based model is that we cannot be very certain about the toxicity levels. So to understand this and to decrease the chances of toxicity, we have to have some sort of rule-based model on the base of a model. Rule-based designs on the base of a model. These are called as model-based designs. So what we are doing here is we are imputing this kind of the probability of having the adverse drug reaction in a patient. In the computer software, and this probability is basically is a Bayesian probability, and at each dose level, we will try to impute this data into our software, and we'll try to find out what is the what should be the dose of the next level. So this is how it starts. We'll start with the lowest dose. We'll give it to one patient, and we'll increase it until we get the DLT. Once we get the DLT, at each level we are measuring the probability. So once we get the DLT, we'll try it in the bigger cohort, and then we'll come down, and this will be the recommended dose for this drug. Now, the second part is, if we want to have this overdosing computational probability as well, so we will do this overdosing protection at each level. What it means is that at each level, with the probability, Bayesian probability of having of computing the next dose, will also have a protection, so that it will decrease the overdose, decrease the dose such that the overdose will not happen. So this will again be happening at each level. The advantage of having a escalation with the overdose control is that we will have less number of side effects or less number chances of serious toxicity in the patients who are giving the dose. So the end result of this whole exercise is basically to give a phase 2 dose. Now, if you can see in this pembrolizumab, there was a cohort A1. The first cohort was given 10 milligram dose and in the 7 patients. And secondly, again, the cohort was increased and A2 cohort was given 13 in the 13 patients. So that was the dose phase 1 trial for pembrolizumab. So after that, we come to the next phase, which is the phase 0. Phase 0 studies are basically microdosing studies. The FDA says that we should be doing, the FDA actually encourages the companies to go for the phase 0 studies because these are very small. These studies can be performed very easily and in a very small number of population. So what it means is that we have to establish the dose range in the future studies. So for establishing dose range, we are getting a very small dose. So we can do this with a very small number of very small preclinical data, very limited preclinical data. But the issue is that we have to have a very good biomarker. So because we are getting a very small number of small amount of dose, the biomarker has to be very robust in this case. So in preclinical studies, most of the times we will do pharmacokinetics, but pharmacodynamics with the if there is a marker which can be assessed at a very low dose and modulation can be targeted, then pharmacodynamics should also be done at the phase 0 level. Very less number of patients, around 10 patients are required for this, and usually it is a 1 day study, and it will not go beyond 7 days. It's a single cycle study, and the advantage is that multiple targets, multiple drug candidates can also be needed from this phase 0. Now, typically, it will take around 3 months, 3 to 4 months to complete this study. If we see the design of this study, we can see that we have a pharmacokinetic analysis at day 0 and 2, 4, 7, and 24 hours after giving the dose. Then pharmacokinetics of urine can be done at 0 and 24 hours. And for pharmacodynamics, tumor biopsy is done usually at a time when the drug is matched, the Cmax is matched with the tumor biopsy, and we usually stop at before 7 days. Should not be more than 7 days. So what it provides us is a decision making whether we should go for the phase 1 trial or we should not go for the phase 1 trial. So basically, it is if the pharmacokinetics and dynamics are good, we can go for the trial. If it is not good, then we should not go for the trial. So in case of polymerase inhibitor ABT888, the proof of concept was done and phase 0 trial was also determined. What it produced was, firstly, it gives a real-time assessment of pharmacokinetics and dynamic studies, as well as predefined assay technique was used. There is an issue of tumor biopsy consent because we are doing a biopsy of the tumor without any therapeutic benefit. We have to ask the patient that there will not be any therapeutic benefit, but it's just to do pharmacodynamic and kinetic studies. Now, just a word about the historical controls. These are basically for the very rare kind of cancers in which there are not substantial number of patients. So we can have the control group from the in which the placebo is given or any other drug is given. That control group is the historical control. That means that has been done in previous studies, and we are taking that group as the control for our study. Now, there are many issues with that. The major issue is that the drug has to have a very predictable course, and the endpoint should be very objective, so that the endpoint which is measured in the previous studies should be very similar to the endpoint that we are measuring. The issue is there is a lack of randomization, there is a lack of blinding that is there, but major major drawback of this is that there is a confounding factors. So we cannot control all the confounding factors. We can never control all the confounding factors, and that is the main issue with the historical controls. Now, if we take a look at the classical designs for phase 2 and phase 3, let's see there we have patient groups with the tumor of lungs, liver, and kidney. So out of this, what we are doing in a classical design, we'll take all the tumors which are similar, like in case of lung tumors. Then we'll give in the trial one, we'll give this type A kind of tumors with drug A, and we'll assess the endpoints. This is the classical design. So again, we'll do it. We'll do the same for the liver tumors, the brain tumors. That is what we are doing in the classical design. But the problem here is that we know that in cancer there are multiple mutations. So, for example, the orange mutation is there in all other cancer types, and we know that most a lot of the newer drugs which are coming to the market, they work on the basis of mutation. So there's a chance that this mutation the patient of this mutation may get cured, but the other mutations which are blue and green mutation will not be cured. So and, after that, what we get is a very haphazard results which will not make any sense, or the drug may go into clinical failure. So therefore, new designs are created. What it means is we want to go for multiple different drugs so that we can try on different kinds of different kinds of. So this is called as master protocol. What do you mean by master protocol? The master protocol is basically the combination of many different protocols into one protocol. Why we want to do it? Because we want to have multiple drugs, and also we can have the common placebo control. Why we want to have common placebo control? Because we want to give most of the drugs to the patients. We don't want to give placebo to the patient. So therefore, a master protocol is now a very good idea for doing the trials. So how do we go about master protocol? Master protocol is used in platform trials. There are two kinds of platform trials that are available, that are usually done. One is the basket, and the second one is the umbrella trials. So in basket trials, what we do is we take the mutation of all the similar mutation, and then we'll go with of all different kinds of cancers, and then we'll treat it with a single drug. So we'll do the same for other drugs. So what we are doing is we are creating a basket with different kinds of cancers, and once that basket is there, we are treating it with a single drug. So, for example, in the pembrolizumab patients, we are treating breast cancer, we are treating ovarian cancer, we are treating solid neoplasms, and we are treating locally advanced tumors. All these are being treated with the single agent, which is pembrolizumab. This is a type of a basket trial. Second one is the umbrella trial. What is an umbrella trial? Is just opposite of the basket trial. So here we are using many intervention and one kind of cancer. So we are using lung cancer here, And if the lung cancer is there, and we are, this is the basically the whole umbrella is the disease. So we are taking one disease and we are giving different kind of the spokes of this umbrella are the therapeutic arms. So we are giving multiple treatments for a single kind of cancer. For example, pembrolizumab was again used for non-small cell lung cancer, advanced lung cancer, and there are many combinations. First, the pembrolizumab was used with the combination of with the other chemotherapy. It was used alone with PD-L1 positive patients, and it was again used alone in the non-small cell lung cancers, which were previously treated with anti-PD-1 therapy. So this is kind of an umbrella master protocol. So just to recap, if we are using multiple diseases and a single intervention, it's a basket trial. If we are using single disease with multiple interventions, it can be a umbrella trial. So what we have here as a platform is that what we have here is that we can do an interim analysis. Interim analysis is nothing but a small analysis which has to be pre-specified in the protocol at what time and who will do the this interim analysis. And at this time of interim analysis, what is new is that we can continue some arms which are performing good. We can drop some arms which are not performing good, and we can also add some arms if we deem it necessary. We can also add this arm, and then there is a final result. So we have a platform here from which we can launch or we can drop some arms. This is called as the platform trial. This is also called as the adaptive clinical trial because we are adapting to the situation. We are adding arms and we are also removing some arms. So a few words about the N of 1 trial. N of 1 trial, as the name suggests, is we are only having one patient here. And we are randomizing if we are giving two drugs, let's say A and B, we are randomizing the series of that drug, how it is given. So in this case, the drug A is given, then we'll assess the endpoints, then a washout period is given, then again drug B is given. After that, we assess the endpoint, and further we can have a randomized list what other drugs can be given. So the very important point is that after each assessment, before the start of giving the second drug, we have to take consent from the patient whether we should go about or not. In cancer chemotherapy trials, why they are used in cancer chemotherapy trials is because in the pediatric age group, we have to have a lot of experience for these patients with a particular set of drug. Secondly, if there is a very rare cancer or very rare disease, in that case also we have to use N of 1 trial. So this will give us the best evidence for a very less number of patients. These are the other designs for the trial. These are the other designs for N of 1 trial. So next is the expedited approval programs of FDA. So FDA has 4 programs for expedited approval. The 3 are the designations, and 1 is the approval pathway. So the first one is the fast track program or fast track designation. It was introduced in 1988. Why it was important is that it was creating a new treatable population. So what it means is that it is used for all the programs or for the serious conditions. But this program was important for the unmet need. That means there was no drug for that illness. So the situation is very desperate situation, and therefore non-clinical data can also be used in this kind of trials. So at this point of time, there were 0 patients under treatment. And what the fast track program or what the drug is doing is it is increasing the number of patients under the treatment. So it has to be done within 60 days. The FDA has to give the answer, and IND has to be filed, and it is usually done with the IND or after the IND. Then what it gives is the rolling review. Now, what is a rolling review? What is a review? So the FDA or any other agency, it requires all the data in one go. So we have to give the efficacy data, we have to give the adverse drug reaction data, all this data in one go, chronic and acute toxicity studies data that has to be given in one go. But in case of fast track, in case of rolling review, what we can do is we can give the data piecemeal. So if our efficacy data is completed, so we can give that data to the FDA. If our toxicity data is complete, so we can give that data to the FDA. The FDA will do very early feedbacks, will give very early feedbacks, which will help the drug development process. And FDA will also give frequent meetings also. So this is the fast track. The second one is the accelerated approval. This is the pathway, the approval pathway. So it is also for the serious condition, but we have to have an existing drug, and we will use it with the help of the surrogate markers. So what is a surrogate marker? We have already discussed. So why the surrogate markers are used is because that they will decrease the time period for doing the clinical trial, and therefore this is important. It can also be filed, and it will what FDA will give is a preliminary approval. So before even the confirmatory studies by the clinical endpoint. The third one is the priority review. It is also a designation. It's also for the serious conditions with the advantage over the existing condition. In this, usually the review time for the FDA is 10 months. So in the priority review, this time is decreased by 4 months to just 6 months. So there is a shorter period of time for the review process of the NDA. It is usually filed with the NDA or afterwards, and the FDA has to give the answer within 60 days. And the last one is the breakthrough therapy. The breakthrough therapy is basically the fast track therapy, but here that the drug is already present. So we are increasing the number of the patients which can be treated effectively with the help of the newer therapy in question. So it will give all the features of the fast track designation, which is rolling review, intensive FDA guidance, and this has to be filed with the IND or after 60 days. So to summarize, the phase 0 is usually done with the FDA is now requiring to use the phase 0. It is usually done for less than 1 day. The phase 1 studies, these are the dose escalation studies, and there are 2 kinds of studies, which is rule-based and model-based. Definitely model-based is much better because it decreases the adverse drug reaction or toxicity happening in the patients or in the healthy volunteers, but it requires software. The phase 2 and 3 designs in oncology is basically a basket and umbrella design, which are adaptive clinical trial and with the master protocol. The N of 1 studies are a single patient studies, and it is used for very rare diseases and to gain the experience in one patient. And finally, the FDA has a few programs like fast track, breakthrough therapy, accelerated approval program, and priority review. So with that, if you have any questions, please do ask.
Thank you so much, sir, for the insightful presentation. We will take up a couple of the questions from the delegate side. I will also request the participants to keep posting the doubts, queries in the meantime. So the first question that we have got is, when should the phase 0 should be initiated, sir?
So the question is, when should be the phase 0 should be initiated? So basically, phase 0, the FDA says that before the completion of the phase 1, or even before the start of the phase 1, we can start the phase 0 trial. We have to take the permission of the FDA. You can also do the phase 0 trial at the initial part of the phase 1 trial. So when you are starting the phase 1 trial, you can have that phase 1 trial. But ideally, we should complete the phase 0 trial before the start of the phase 1 trial.
Thank you, sir, for answering the question. We also have another question from the delegate side. They have asked the Indian guidelines for fast track programs, sir. Please elaborate on that.
Okay, why we have taken that slide of faster programs, not the Indian guidelines, we have taken the FDA guidelines, is because we don't have a much extensive guidelines how to do fast tracking or how to do breakthrough fast tracking. So all these are not not available in the even in the NBCT rules. Only thing is that they have mentioned the fast track approach, but how to go about it, this is missing. What are the benefits that are there? How do the companies get benefit of fast tracking that a drug? So that kind of data is or that kind of procedure is missing in the NBCT. Therefore, we took the programs of the FDA.